Biochemistry examines the different biomolecules. Remember that the biomolecules are proteins, lipids, carbohydrates, and nucleic acids. In this lecture, we're going to examine fatty acid metabolism. At a later lecture, we'll also examine the synthesis of cholesterol. This lecture focuses on palmitate, the central fatty acid used by the cell. Palmitate can be modified into other fatty acids and is therefore a good molecule to examine for catabolism or breakdown and anabolism or synthesis. Both processes will be covered. You will know you are finished with this material when you can do the following without your notes or book. Describe the general structure of fatty acids. Explain the mobilization of triacylglycerols. Describe the beta oxidation pathway and the chemical reactions. Describe the palmitate synthesis pathway, and discuss regulation. Some of this may be somewhat confusing. Just be aware, with practice, this material can be learned and ties into other material in this course. Before we begin, it's useful to have a brief overview of the concept of the pathway. No doubt you've heard of glycolysis and the tricarboxylic acid cycle, aka the T8-TCA cycle. These prepackaged systems are referred to as pathways. We've looked at enzymes already. As you now know, an enzyme is a protein that performs catalysis of chemical reactions. Okay, that's fine. However, it's important to realize that enzymes do not work in isolation in the cell. Each enzyme, except for a few, takes in a substance created by some other enzyme. In this way, a network of chemical reactions is established. So enzyme A, for example, modifies a substrate and passes that material on to enzyme B. Enzyme B modifies it further and passes the result to enzyme C, and enzyme C modifies it further and passes it to enzyme D, and so on and so on and so on. Pathways come in a variety of networks, including linear systems, cycles, and complex exchanges. The figure shown here is a general pathway map for cellular metabolism. As you can see, the pathways themselves are not in isolation, but are connected to other pathways. One of the big keys is that pathways that require energy, typically anabolic pathways, are tied to pathways that release energy, typically catabolic pathways. In this way, energy transfers occur that keep the system going. Lipids and fatty acids are primarily hydrophobic molecules. They include triacylglycerides, phospholipids, cholesterol, and so forth. They're used in a large variety of biological functions, including membrane formation, signaling, solvents for hydrophobic materials, and even maintenance of tissues such as those found in the lung. Whether fatty acids are being stored or released, the flux of fatty acids is controlled by the current state of the organism. That is, whether the organism has eaten recently and is fed, or the organism has not recently eaten and is fasted. The general fatty acid is a simple structure. It possesses a carboxylic head group attached to an alkyl chain. This chain is referred to as the tail, and it can be either saturated or unsaturated. If the chain is saturated, then every carbon has a full complement of hydrogen bound. If the chain is unsaturated, then there are one or more double bonds in the chain where carbons are missing hydrogens. The examples shown here are palmitic, or palmitate if it's the conjugate base, which is a fatty acid with 16 carbons. Also shown is cis-palmitoleic, or cis-palmitolate, is if it's a conjugate base, which is the same fatty acid with a cis double bond. The carboxylic head group can be modified with attachments, and the alkyl chains can be of varying lengths. The presence of the carboxylic head group makes these amphipathic, or capable of interacting with water and hydrophobic solvents. The head group is polar and will interact with water. The tail is hydrophobic. So how big are these things? Well, they can be pretty darn big. Palmitate is 16 carbons long and forms the foundation of fatty acids for others. Here are two examples showing how long these molecules can get. Nervonic acid is 24 carbons long, and all cis 4, 7, 10, 13, 16, 19 docosahexaenoic acid is 22 carbons long. So you can see, big. Most fatty acids are about 16 to 20 carbons. Frequently, you'll see 14 carbon long chains linked to proteins, especially lipid-anchored proteins, whereas the really long ones are found in complex lipids in the nervous system, such as the docosahexaenoic acid we saw previously. Just by way of review, here are some additional key fatty acids that are found in metabolism. Propionic acid is used in gluconeogenesis and metabolism of odd chain, meaning odd number of carbons, fatty acids. Palmitate, this is what we are examining today. Palmitoleic acid 16,1 with one double bond is the most common fatty acid in triacylglycerols. Linoleic and linolenic are essential fatty acids, meaning we cannot make these efficiently enough, so they must be gotten from diet. The fact that these molecules are amphipathic and have substantial hydrophobic character is very important. We already know about the creation of membranes which define the limits of the cell, as well as making the compartmentalization we see in cells. However, there are other traits as well. First, these molecules do not require a lot of water for storage. Glycogen, which is a polymer of glucose, requires a tremendous amount of water for storage. With triacylglycerides, there's potentially unlimited storage of these fuel sources. Gram for gram, these molecules provide about 2.5, maybe a little bit more energy than glycogen, and they work great with temperature regulation. So as homeotherms, we maintain a constant temperature, they are an ideal molecule. When talking about fatty acids, we want to start with the triacylglycerides. This is a primary fatty acid carrying molecule, as is the main form of storage for fatty acids. Triacylglycerides form adipose tissues and can be found under the skin as subcutaneous or around organs, which is termed visceral. Structurally, a triacylglyceride is a glycerol moiety substituted with 3 fatty acids. Each fatty acid can be removed from the glycerol backbone by a class of enzymes called lipases. Here is the typical structure for a general triacylglyceride. As you can see, the top of the molecule is the glycerol backbone, and there are 3 attached fatty acyls, 2 of which are unsaturated. This molecule is not very amphipathic and is going to be primarily hydrophobic. This structure makes it easy to see why this molecule is good for storage, as 3 fatty acids are attached to it. Each one of these fatty acids can be removed by a lipase and then oxidized down to release energy and create acetyl-CoA. This quick reaction shows how lipases remove fatty acyls from the triacylglyceride. Lipase uses water to recreate the carboxylate on the fatty acyl, and the intact glycerol remains. In this image, the glycerol shown as having chirality, which it doesn't. Glycerol is prochiral, meaning that it becomes chiral when substituted, but by default it is achiral. You should also notice that this event releases protons, which could affect the pH of the system. This is a very common motif for metabolic events. The fact that we're breaking down a material means this is a catabolic reaction. To get energy from fatty acyls, there are 3 steps that need to be performed. First is the mobilization of triacylglyceride from adipose tissue, though this can be avoided by using triacylglycerides in the diet. Next, the fatty acyl must be removed into the matrix of the mitochondria. Lastly, the chain is oxidized down to acetyl-CoA, and some energy is created in the process. Acetyl-CoA is our key metabolic intermediate. It is used in a huge array of metabolic systems. For our purposes, we will focus on acetyl-CoA as a source of creating energy in the TCA cycle and in anabolic systems where materials are being created. The first step is to activate stored triacylglycerides from adipose tissue. This requires activation of a G-protein coupled receptor system. Please see the supplemental lecture for details on how GPCRs work. I'm not going to cover them here, but I have posted a secondary lecture to provide those details. Hopefully you took a moment to review the GPCRs or already understand the G protein coupled receptor system. Fatty acyl mobilization is controlled by a GPCR. Here we see a hormone such as glucagon binding to a 7-transmembrane receptor that activates a G protein. The alpha subunit of the G protein activates adenylate cyclase, a very common downstream target, that converts ATP to cyclic AMP. The cyclic AMP activates protein kinase A, which in turn phosphorylates and activates triglycerol lipase. Notice this enzyme is activated by phosphorylation, so we expect certain amino acids to be available for phosphorylation, and mutations at those amino acids would result in failures of this enzyme to activate. The active triacylglycerol lipase converts triacylglycerol to diacylglycerol, and other lipases remove the remaining fatty acyls. What's left is glycerol and free fatty acids. The free fatty acids can be exported to the blood and ultimately transferred to mitochondria in needing cells. So in adipose, we're talking about transferring fuel out of storage and making it available to the body. Hormones such as glucagon and epinephrine will activate the 7-transmembrane receptor, which results in activation of hormone-sensitive lipase. Additionally, perilipin, this is a small protein, is also phosphorylated by protein kinase A and reveals the stored fats. The lipase has access to the fats and removes them from the glycerol backbone. Fats are carried by serum albumin, where they go where they're needed, while glycerol is uptaken by the liver. In the liver, the cells do this trick. Glycerol is converted to L-glycerol 3-phosphate by glycerol kinase, and we can see the addition of phosphate to the structure. The next enzyme in the path, remember a pathway is a series of linked enzymes, is glycerol phosphate dehydrogenase. This enzyme oxidizes the glycerol phosphate and creates NADH, an electron carrier, and the product is dihydroxyacetone phosphate. The next enzyme, triose phosphate isomerase, converts dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Pay very close attention to these two molecules. You'll see them again as they are used in glycolysis. Here's a general overview of what's going to happen. We've already seen part of this. In adipose, a triacylglycerol is broken down to glycerol and free fatty acids. The glycerol goes to the liver to either become pyruvate via glycolysis or glucose via gluconeogenesis. We'll see those processes later. The fatty acids, however, will go through beta oxidation, called here fatty acid oxidation, to become acetyl-CoA. This molecule is used by the tricarboxylic acid cycle, termed here CAC, to produce electron carriers for energy. An acetyl group is 2 carbons, so if palmitate is 16 carbons, we should anticipate that 8 acetyl groups can be made from 1 palmitate. Each of those acetyl groups provides NADH and FADH2 in the TCA cycle, and those electron carriers go to the electron transport chain to make energy. As you can see, a triglyceride with 3 fatty acyls and a glycerol can make a lot of energy. A fatty acid on its own cannot enter a mitochondrion. It needs a carrier. The carrier is called coenzyme A. This is the same carrier of acetyl-CoA. In this case, we'll call the product acyl-CoA as opposed to acetyl-CoA because the chain is longer than 2 carbons. This reaction is accomplished by the enzyme acyl-CoA synthetase. Notice the reaction shown is being freely reversible, maybe even equilibrium, though we don't refer to equilibrium in cells. A free fatty acid arrives at the cell. The enzyme acyl-CoA synthetase, which is located on the surface of mitochondria, binds the fatty acid to coenzyme A. This requires the consumption of ATP, making AMP and pyrophosphate in the process. This reaction is very nearly at equilibrium. Essentially, it is at equilibrium, but we know this would not work in the cell. So how does the cell prevent the reverse flow of this reaction? We'll use pyrophosphatase to cut the pyrophosphate. Remember, an enzyme needs all of the substrates of the reaction to work, and all enzymes can run backwards. However, by removing the pyrophosphate, this reaction cannot flow backwards and is instead driven forwards since a product, pyrophosphate, is being depleted. Preventing backflow by splitting pyrophosphate is a very common motif in metabolism. Additionally, by removing a product, i.e. the pyrophosphate, the reaction is actually driven forward. Now that we have the carrier, it must move into the mitochondria. The acetyl-CoA binds to the enzyme carnitine and transfers the acetyl. There are two carnosine-acyl transferases, 1 and 2. 1 is on the cytoplasmic side of the membrane of the mitochondria, and 2 is on the matrix side. Transfer of an unacylated carnitine drives import of acyl carnitine. Here is the system. First, the energy of acetyl-CoA allows for transfer of acyl to carnitine. The enzyme is carnitine acyl transferase 1. The acyl carnitine is moved by the enzyme translocase to the matrix side of the membrane. Next, the enzyme carnitine acyl transferase 2 moves the acyl off of carnitine and back onto CoA. Yes, it's odd. We add something to CoA, then pull it off, transport, and put it back onto CoA. Metabolic pathways do this frequently in order to get around membranes. As a note, the export of carnitine via translocase provides the energy for the import of acyl carnitine. Now that we have the acyl-CoA molecule located in the matrix of the mitochondrion, we have gone from adipose tissue, released free fatty acid, and transferred it onto a CoA and moved it into the mitochondria. Next, we degrade it. The beta oxidation pathway is an energy-preparing pathway. The goal of the pathway is to make three materials. Acetyl-CoA, this is the input material for the TCA cycle. NADH, this is an electron carrier used in the electron transport chain. FADH2, this is another carrier used in the electron transport chain. Here is the beta oxidation pathway. It looks complicated, but it really isn't. Overall, we're going to use 4 enzymes to reduce an acyl-CoA by 2 carbons and make an acetyl-CoA and an acyl-CoA. That is 2 carbons shorter. This process is iterated until the fatty acid is completely degraded to acetyl-CoA. First, the acyl-CoA is oxidized. Notice the creation of the trans double bond. This produces trans delta 2 enoyl-CoA and FADH2. This enzyme is performed by the enzyme acyl-CoA dehydrogenase. Notice the enzyme names tell you what they are doing. This is very useful in biochemistry. Next, enoyl-CoA is hydrated by enoyl-CoA hydratase. This removes the double bond and we now have L3-hydroxyacyl-CoA. Our next step is another oxidation, this time to make a ketone. The enzyme is L3-hydroxyacyl-CoA dehydrogenase. Again, notice the enzyme name. Enzymes are very frequently named for their substrates and what they do to the substrate. Another product of this reaction is NADH, an electron carrier for the electron transport chain. We now have 3-ketoacyl-CoA. Notice the ketone. We have everything ready to go. We're going to remove the rest of the chain, leaving the acetyl-CoA behind. The remaining acyl is added to a CoA molecule. This is a thiolysis with the sulfur picking up the fatty chain, and what is left behind is acetyl-CoA. The enzyme is beta-ketothiolase. Look carefully at this structure. We started with, say, palmitate, 16 carbons, and prepared a chain of 14 carbons to be removed. By removing the 14 carbons, we left behind an acetyl-CoA and created another acyl-CoA with an acyl chain of 14 carbons. This may be slightly counterintuitive, so practice this pathway and structure logic until you're comfortable with it. Now that we have seen how to break down a fatty acid, our next step is to build one up. The goal of fatty acid synthesis, for our purposes, is to create palmitic acid or a chain of 16 carbons. Most anabolic pathways where we are building a material involve the creation of some building block that gets used over and over again. Palmitate is no exception to this. Our building block is acetyl-CoA. which is found in the mitochondria. So yes, it is possible to degrade a long-chain fatty acid to acetyl-CoA and to use that acetyl-CoA to build up palmitate. That's a cell for you. As you'll see, the first metabolic destination of acetyl-CoA in the mitochondrion is to become citrate. Acetyl-CoA is combined with a molecule called oxaloacetate in the first step of the tricarboxylic acid cycle and becomes citrate. The enzyme that does this is named citrate synthase. Citrate has several metabolic destinations, only one of which is TCA. In this case, citrate is exported from the mitochondria and is cut back into acetyl-CoA and oxaloacetate. We saw this before, where a molecule is modified, transported, then restored. Look at carnitine if you've forgotten. The acetyl-CoA is converted to malonyl-CoA. Now that's our building block. Now acetyl-CoA plus malonyl-CoA combine to create a 4-carbon fatty acid. This process is repeated with malonyl until the 16-carbon chain is finished. Let's take a closer look. This part is really the most complicated part of the process. The actual creation of the chain is a stepwise linear process. This part, however, is a complex dual pathway, so study it carefully. It begins with transporting citrate out of the mitochondria. Remember, citrate is synthesized from acetyl-CoA and oxaloacetate. Here we are. Look towards the bottom of the figure and you'll see citrate. Remember, palmitate is 16 carbons long, so we're going to need 8 acetyl-CoA, since each acetyl is 2 carbons. So 8 times citrate is exported from the mitochondria. Here's what happens. Citrate in the cytosol is cut by ATP citrate lyase. This consumes an ATP and releases acetyl-CoA and oxaloacetate. The acetyl-CoA is used immediately in palmitate synthesis in this case. Oxaloacetate is converted by malate dehydrogenase to malate. This consumes NADH and is a reverse reaction to what is seen in the TCA cycle. Malate is converted to pyruvate by malic enzyme. Pyruvate is a glycolysis product and NADPH is made in this reaction. The NADPH, notice this is not NADH, is used in palmitate synthesis. As a result, we get our 8 acetyl groups and 8 NADPH needed for reducing capability in palmitate synthesis. We're now mostly ready for synthesis. We have the 16 carbons in the form of acetyl-CoA. We have most of the NADPH needed. This came from the malate to pyruvate reaction. The other 8 NADPH come from a different pathway we're not covering, but is covered in metabolism classes named the pentose phosphate shunt. Just as a reminder, metabolic pathways are deeply interconnected. In this pathway, we've seen glycolysis and TCA provide some materials. We see a side reaction providing reducing equivalents, and there is another pathway called the pentose phosphate pathway, sometimes called 3P, or the pentose phosphate shunt, provide reducing equivalents. The electron transport chain will provide ATP. This is a simple graphic just showing the relationship of various pathways in connection with palmitate synthesis, listed as fatty acid. You'll see glycolysis, TCA in the mitochondrion, and the pentose phosphate pathway. Now that we have materials and reducing equivalents, we're ready to begin synthesis. The step shown here is the first and key regulatory step of the pathway. The rate of this reaction controls the rate of the entire synthesis. Acetyl-CoA combines with carbonate and ATP using the enzyme acetyl-CoA carboxylase to make malonyl-CoA. Notice that malonyl-CoA is the same thing, just with the carboxylate group added. Water is going to be a leaving group in this reaction. Notice the terminal methyl of the acetyl loses a hydrogen, as does the carbonate. Also, carbonate loses an oxygen, so there's your water. Additional products are ADP and inorganic phosphate. So the phosphoryl hydrolysis is providing energy for this reaction. Malonyl-CoA is our building block. The first elongation to 4 carbons using an acetyl-CoA and a malonyl-CoA is a little bit different than further elongations. Just remember, in this first case, we're adding malonyl to acetyl. In future elongations, we're adding malonyl to the growing chain. Acetyl and malonyl-CoA both bind to a protein called the acyl carrier protein. They undergo condensation reactions to form the product acetoacetyl ACP, acyl carrier protein. Notice the CoA is lost. The transfer off of the CoA provides the energy. The thioester bond in CoA is a high energy bond. The enzyme that does this is acyl malonyl ACP reducing enzyme. Notice it's called acyl, not acetyl. In future steps, the malonyl is attached to the growing acyl chain. Only on this first step is it acetyl. Now that we have the 4-carbon chain on the ACP, we need to make it a proper chain. The rest of these steps do just that and prepare the chain for the next malonyl. We're at acetoacetyl, and it's reduced by NADPH to D3-hydroxybutyryl ACP. The ACP is just the carrier. The enzyme is named beta-ketoacyl ACP reductase. Next, a water is split out in a dehydration reaction to make crotonil ACP. The enzyme is 3-hydroxyacyl ACP dehydratase. Crotonil ACP is reduced to butyryl. Notice the name is butyryl. This is 4 carbons long. Remember butane? The enzyme is enoyl ACP reductase. From here, we repeat all the steps by adding another malonyl. The malonyl is going to connect to this butyryl rather than the acetyl, as I mentioned. This process continues until a 16-carbon ACP is formed. Once the chain is this long, an enzyme named thioesterase cuts the C16 from the ACP. This enzyme is part of the mammalian superenzyme and is specific for 16-carbon chains and won't cut shorter chains. So the synthesis part of this pathway lists several enzymes, except they aren't really separate enzymes. In this case, all the enzyme activities are found on one superenzyme called the mammalian fatty acid synthase. There are 7 active sites on one peptide chain. This is a huge enzyme complex made from 2 dimers. Each monomer has all the functions, but only the dimer is actually functional. Here are a couple images of the synthase just so you can see the full enzyme system. In the cross section, if you look carefully, you'll see the word pivot. This is the ACP part, and the chain simply swings from one enzymatic activity to the next. We've looked at 2 processes, oxidation and synthesis of lipids. Obviously, cells don't allow all systems to run at full speed all the time. In lipids, there are basically 2 major states of regulation, fed and fasting. After eating, dietary lipids are stored as triacylglycerols. Additionally, anything beyond what is needed for energy or protein synthesis will also be stored. This goes for carbohydrates and amino acids. At this time, insulin is activated. This is the hormone that is released when you eat, and insulin triggers increases in malic enzyme as well as pentose phosphate shunt enzymes. So all of this together works to increase palmitate synthesis. Remember, we are likely breaking down some fatty acids to create palmitate for storage on triacylglycerides. At the same time, hormone-sensitive lipase is decreased. Remember, this enzyme releases fatty acids from triacylglycerides. During fasting, we see dramatic changes in regulation. First, there is loss of glucose as it's used up, and insulin also decreases. As a result, glucagon increases, and remember, glucagon activates the G protein system for fatty acid degradation. As a result, we get more free fatty acids to the blood that are uptaken by cells, and consequently more energy production and acetyl-CoA production through beta oxidation. As a brief summary, we talked about lipids and their structures and activities, as well as hormonal control over lipid uptake, synthesis, and degradation. We've also talked at length about fatty acid synthesis and how it is connected to other metabolic pathways. So there you have it, palmitate metabolism as performed biochemically. Be sure to focus on memorizing the pathways and being able to explain chemically what is happening in those pathways. Your book has further diagrams to help with this. During the work days, you'll be practicing the pathways as well as answering questions from your book and other questions. Take your time, and welcome to Metabolism.